Hello everyone, welcome back to Beard Biology. Today we will discuss about the fluid mosaic model. We know there is so many hypotheses describing the structure of biomembranes, like sandwich or trilamellar model, unit membrane model. But among them, the most accepted and the most latest model or hypothesis is the fluid mosaic model. Today we will discuss about the fluid mosaic model. This model is given by Singer and Nicholson in the year 1972. This model is given by Singer and Nicholson in the year 1972. According to fluid mosaic model, proteins are arranged in phospholipid layer as mosaic pattern. Thus, membrane is termed as protein iceberg in a sea of phospholipid. Thus, the membrane is termed as protein iceberg in a sea of phospholipid, or gulab jamun in concentrated solution of sugar. That means gulab jamun is the protein and the concentrated solution of sugar is compared with the phospholipid. Gulab jamun in a concentrated solution of sugar. We will discuss in two parts the fluid mosaic model. One is the phospholipid and another one is the protein. We will discuss about two parts, the phospholipid and the protein. The phospholipid and the protein. At first, we will discuss about the phospholipid. The phospholipid is the main component of the cell membrane. Phospholipid is the main component of the cell membrane, that is the major component of the cell membrane. This phospholipid layer is arranged in bilayer. Where we find that the phospholipid molecules of a phospholipid molecule, the polar head that is hydrophilic in nature is situated towards the outside. Polar head that is hydrophilic situated towards outside, and the nonpolar tail of the phospholipid molecule that is hydrophobic in nature is situated towards the inside. This ensures that the nonpolar tail of saturated and unsaturated hydrocarbons is protected from the aqueous environment. Now we find that the phospholipid is rich in unsaturated fatty acid. Phospholipid is rich in unsaturated fatty acid, which is liquid in nature. This unsaturated fatty acid is liquid in nature. So this provides the fluidity to the phospholipid. Which help in cell growth. The fluidity help in cell growth, formation of intercellular junction, endocytosis, secretion, and cell division. Here we find the lipid of phospholipid. Phospholipid is a kind of lipid. In the cell membrane, we find the lipid like phosphoglycerides. This lipid is quasi-fluid in nature. It is the most important thing of the phospholipid layer, that is quasi-fluid in nature, which provide the lateral movement of intrinsic proteins through the overall bilayer, which provide the lateral movement of the proteins through the overall bilayer. Moreover, we find some amount of cholesterol in the phospholipid layer. This cholesterol provides stability to plasma membrane. This cholesterol provides stability to the plasma membrane. But this cholesterol is absent in the plasma membrane of prokaryotes. Cholesterol is absent in the prokaryotes. After that, we will discuss about the protein present in the cell membrane. According to the fluid mosaic model, there is two types of protein present in the cell membrane. One is integral or intrinsic protein and another is peripheral or extrinsic protein. Integral or intrinsic protein and peripheral or extrinsic protein. Integral or intrinsic protein that is tightly bind to the phospholipid layer and cannot be separated easily. Tightly bind to the phospholipid layer and cannot be separated easily. In the other hand, the peripheral or extrinsic proteins are superficially bind to the phospholipid layer and can be separated easily. The integral or intrinsic proteins are termed as tunnel protein. The integral or intrinsic proteins are also termed as tunnel protein or channel protein. This tunnel protein or channel protein actually facilitate the movement of water soluble materials. This tunnel protein or channel protein actually help to facilitate the movement of water soluble materials through the plasma membrane. And in other hand, the peripheral or extrinsic proteins are oligosaccharides of glycolipids. The peripheral or extrinsic proteins are oligosaccharides of glycolipid and glycoprotein. This oligosaccharides of glycolipid and glycoprotein that is present as the peripheral or extrinsic protein are help in the cell recognition mechanism. Best example of cell recognition mechanism is fertilization. Here is the schematic diagram showing overall fluid mosaic model. You can just pause the video and take a screenshot. After that, you can go through anytime a quick revision through this photo. I wish it will definitely help you. If you like the video, please share this video and don't forget to subscribe this channel. Thank you so much.